I'm basically from Sri Lanka. If you don't know where the is, it's just under here. It's a really small island, nice island, which few of you have might have visited. So I'm from Institute of Biochemistry, Molecular Biology and Biotechnology, which is part of University of Colombo. It's part of the main campus, and we are the uh, premier institute in molecular biology in Sri Lanka itself. So to start off, the institute was uh, funded by Swedish grant, and it was started by late Professor Erik Karunanayake, who was sadly passed away this year. He's known as the father of molecular biology in Sri Lanka. So the project that I'm going to talk about started by Professor Eric himself, but not in genomics, but he was more into molecular biology. And the supervisor was Professor Kamini Tendakon, which was a incumbent director of the institute. And the other group member is the first speaker, Professor Eric. We talk about that later on, how everything happened. So my presentation is going to be in two parts. One is I'm going to talk about my project, and the second one, how it happened, how it was made possible. So talking about the project, it's basically about lymphatic filariasis. I don't know whether some of you might have heard about this. It's a tropical disease which is caused by, it's a bit disturbing, yes. Tropical disease that is caused by a nematode called Wuchereria bancrofti. And yes, which constitutes 90% of the infections. The rest of the 10% is Brugia malai, which is Brugia species, again nematodes. So this affects mostly people from underdeveloped countries, would say. If it takes Sri Lanka, we have officially, taken care of the Wuchereria or the filariasis. But if you take African countries, they still have this disease. So basically, 863 million people are at risk. This is statistics from 2020. It doesn't actually kill the person, but it causes, as you can see, really bad disabilities. If you take the WHO has named this as the leading cause of disability in humans. So moving on, the challenge was to find drugs to cure this disease. The WHO currently has a program which it is called Mass Drug Administration, sorry, MDA for short, which is giving a cocktail of drugs, which is three main drugs. But the problem there was it doesn't. One thing is it doesn't affect the male, the adult worm that is living inside the host. So that's one problem. The second was it has developed resistance to these drugs. So the development of drugs again came up. So that's basically what we are trying to do right now. So the next problem is when you try to find drugs, one thing you need to culture them in a lab environment or in vitro. But the problem, there's two problems. One is procuring the verb. taking it out from the host is very difficult to culture in the on the lab itself. But again, culturing in the lab is not possible with this feature area. So, uh, so we needed another way of finding drugs or testing drugs in vitro. So that's where where Setaria, the genome that we have in sequence, come in. So this for decades, this Setaria has been used for to test drugs for lymphatic filariasis. This is a parasite, and again a nematode that is living inside cattle, not in humans. So one, it solves the problem of procuring samples to test. So we can get worms easily from arbitraries, which you can find everywhere, and this can be cultured in laboratory environment, which makes it perfect for drug testing. And it has been proven morphological characters and antigenic characters are very similar to visual area bankrafting. So this in a biochemistry or molecular biology aspect, this was used to test drugs. So the central hypothesis is, I'm just going to read out, if Setaria is genetically similar to the other human parasites, it can be a powerful model organism to discover, test drugs in vitro. So that was our central hypothesis. But up to now, we didn't have a genetic model. So the project was launched to have a genetic model to look at the genetic profile of this Setaria. So we can compare, we can find drug targets. Okay? So with this project, we generated a high quality genome assembly using Illumina reads, which are short reads, and nanopore reads. So we assembled the draft genome with the Illumina reads, and we finalized with the long reads from nanoform. And we parallelly sequenced the transcriptome using RNA-seq to find the expression profile as well. So the draft genome assembly is already available in and the final assembly is coming. So we are waiting for the publication. So take a look at the statistics of the final genome. This is the final genome, not the draft. So we found that the genome size was around 80 megabases, and the number of scaffolds is 159, which gives you an indication of the quality of the assembly. It's a good contiguous assembly, and we tested the quality of the genome with the BUSCO, and it gave us a 97% complete. So it's nearly complete assembly, I would say. And so with this genome, second stage was to analyze whether we could use this as a model organism. So we started the validating process. So the first thing we did was to find the existing drug targets. Now, if you look for a model, you need to find whether you have all the genes that are available in the model. Okay? So we searched the internet and we found 18 genes that are published, which are confirmed. And we looked for those genes in the Setaria genome itself. And we found 17 out of 18 genes in the Setaria genome. So that's basically is a good result. So we can test the drugs that have been published on this genome in vitro. Okay. So some of the examples that we was glutamate, I'm sorry, but glutamic gated chloride. That's one of the targets of ivermectin. Ivermectin is one of the MD-approved drugs that is prescribed by WHO for the mass drug administration. So we found the gene there. And the other, I'm just listing four of these. We have more important ones. The other one is calominin, which is involved in fertility and growth, and transglutaminase involved in cuticle development, and BMR1 and WB123, which is diagnosis assays to identify filariasis disease. Okay? So this basically proves that it has the machinery or the profile that we need to prove it as a model. Then we had a look at the metabolism of this Setaria. And one of the important things we found is if you go through the literature, we found that TCA cycle was not complete, as they say. So we were expecting maybe there was no genes there. But when we look at the genome itself, we found that most of the genes are actually there, and the cycle was complete. So it goes to show that the Setaria itself has a much more complete cycle, TCA cycle, than we thought. So this is just one of them we found. So it shows that maybe looking at the genome or the genetic profile itself would be very important. And when we compared the Setaria genome with the free-living nematode, for most famous one is C. elegans, we found that the nutrient metabolism genes are really low in expression. So that shows that Setaria is again living inside the cattle, which is a parasitic worm. So it depends on the host itself for metabolites. Nutrients. Okay, so, and the other thing with the nematodes is some nematodes has bacteria called Wolbachia inside, some of them, but some of them doesn't. So when we look at the Setaria is not doesn't have Wolbachia itself. Wolbachia parasite or the bacteria actually is helping in the spreading the reproduction. Sorry. So it's helping in the reproduction of the worm when it's inside the nematode. So we found that although the Setaria is not really having the Wolbachia itself, but it still has the metabolic pathways that it needs for reproduction. So that shows that we can use this model for Wolbachia free and Wolbachia harboring parasites as well. So the impact is we have evidence that we can use this Setaria genome as a model organism to test drugs, and it could accelerate drug discovery. So I'm going quickly, and aids in research, and you can basically use it as a model organism. So in conclusion, the bioinformatics actually allowed us to turn a cattle parasite into a model which we can use to find drugs in human diseases. And the genetic similarity we have proven with other filariasis parasites. Not here, but we also had a look at other parasitic nematodes as well. So we had a comparative look at all the human parasitic worms that are available on as genomes. That is not here. So we produced a high quality public resource to empower drug development, and the bioinformatics can help in. This shows that bioinformatics can help in developing fight against neglected tropical diseases. So future directives, that's of course one thing is we can test for drugs in vitro. This makes this possible for lymphatic filariasis. This next thing is multimolecular integration. That is, we have now the genome, the transcriptome, and the proteome of the Setaria, which we can do for multiomics data analysis to find pathways of dynamics of the pathways. We plan to do differential expression. For the moment, we have just one expression set, RNA set. So we plan to do different when it's inside the host and it's outside the host. Now, when we sequence the current worm, it was outside the host for about a day. So most of the genes would have been turned off, which is fighting off the host reactions. So we are planning to do differential expression analysis using the transcriptome itself. We are planning to do molecular docking in silico studies when you find interesting drug targets which you can use. And manual calibration we are planning to do. We went through half of it. I think we annotated, curated around 5,000 genes. We want to continue that. So that's what we did. This project itself, coming from a country that this is a developing country. When we started the project, we had only one sequencing machine for the whole country, just not used because it's costly. So this is the picture from the first EMVNet AGM. You can see me at the back, the small guy here. So this is the first year. Yeah, I was, I think, 2 years out of the university, and I came there. And we didn't know much about bioinformatics and how it works. And these people was talking something, things that are very high because we didn't start any. So, okay. So quickly going through. So MNet helped this project in basically in one in networking, the second in making collaborations. When you don't have people or when you don't have the skills or the equipment, we need to network, right? So we need to find collaborators. So the MNet actually organized workshops and conferences every year. I was fortunate enough to go to most of them. And Sri Lanka didn't have any experts in bioinformatics at all. So this was basically done through MNet. I participated on RNSEC and NGS workshops, and that was a pre-training ground for me. So the second is a collaboration. As I said earlier, the equipment is not there, sequencing. Number one is sequencing. Second one, to analyze, we need the computers. So SLU, we formed a collaboration with SLU, was Eric's institute, and we sequenced the genome there. And the analysis was also done in Sweden at SLU using their infrastructure. So this in a national okay. In a national perspective, this is the first study that was conducted in Sri Lanka, genomic study. And the second thing that came out was with this collaboration is we at IBMBB, that's my institute, is the first and only institute that has a master's program in bioinformatics. We don't have a fully fledged undergraduate program as well. So this is very difficult to get, but EMVNet helped us in developing curriculum and finding teachers and examiners, because as I said earlier, we didn't have any experts there. And so this project was it's a global collaboration example. So it's about helping each other out when we don't have experts. So I'll finish quickly. And is the knowledge and the capacity building was done through EMBNet. So it's very important to make networking and find people who are experts. It might not be, because I'm from a biological background. You need to learn computer science, statistics, whatever. So you need to find people and talk to people and learn from each other. Thank you very much.